All right, hi guys. My name is Nick Brocchini. I'm a student at Brown University studying geology biology, and I'm here today to tell you about the work I did this summer at Hatfield Marine Science Center in Newport, Oregon for NOAA Fisheries on a very small crustacean, a planktonic copepod called Pseudocalanus. I know zooplankton aren't inherently interesting to everybody, but they are to me, and the reason for that is that zooplankton are the way that the ocean turns sunlight into animals. So I study geology biology because I'm really interested in the intersection between physical and biological processes at a planetary scale and also at a very regional scale. And if you think about the way that energy moves through the biological system, phytoplankton in the ocean are using energy from the sun to assimilate, raw nutrients into biomass. But then to get anywhere else in the food chain, that phytoplankton biomass is consumed by zooplankton, which then support things like bait fish, larger commercial species, animals that we might not eat but that we think have inherent value, and us. We fish and we eat fish. So zooplankton are critical both to the oceans as we know them and to life as we know it. Obviously, you don't just need sunlight or energy to make an animal. You also need materials, also known as nutrients, and I was fortunate enough to work in a place this summer where the ocean has an abundance of nutrients. That was the Oregon coast. The Oregon coast experiences really strong upwelling, which means that nutrient-rich deep water rises up to the surface into the photic zone where those nutrients can be used by primary producers to produce lipid-rich, nutritious biomass that can be taken advantage of by other consumers, including zooplankton. The upwelling there is interesting because it varies seasonally. It's much stronger in the summer, meaning that there is a summer high production cold water regime and a winter lower production warm water regime. There are also longer than annual cycles like El Nino and Pacific Decadal Oscillation that can impact the strength of upwelling as well. The other great thing about working in Newport, Oregon, where I was, is the proximity to the Newport hydrographic line. The Newport line is a transect that's been sampled routinely since 1996 at stations between 1 and 200 nautical miles offshore. All of those stations are sampled at least 4 times a year. Some of them are sampled much more frequently, including the Sentinel station, NH05, which is sampled twice monthly. And we're going to see a lot of these station codes. The way you read that is 05 means that it's 5 nautical miles off the coast. NH means it's the Newport line. So hopefully I successfully convinced you that zooplankton are important, but maybe I haven't yet convinced you that Pseudocalanus specifically are something you should care about. But you should. There's 2 reasons for that. One is that they're extremely abundant off the Oregon coast, and the other is that they're highly nutritious. They have lipid-rich biomass that helps to transfer a lot of nutrition and energy up the food chain. And they're actually one of 3 northern copepods that are used as one of a suite of ocean ecosystem indicators to assess ocean health. And the northern copepod biomass anomaly in this figure can actually be correlated with salmon returns. But the thing about Pseudocalanus is that it's a genus name, not a species name, and we actually were not 100% sure which species of Pseudocalanus were occurring off of the Oregon coast. Species of Pseudocalanus are really challenging to distinguish from one another morphologically. One of the main ways you can do that is by looking at size and the differences in sizes between species to make those distinctions. But we weren't sure whether size was an effective diagnostic tool or not. We had seen some variation in the size and abundance of Pseudocalanus off the Oregon coast, both with season and also within single samples. And we knew that multiple species had been found both to the south of us in California and to the north of us in Washington. So we wanted to get a better grasp on the variation in Pseudocalanus species and the variation in Pseudocalanus body size off of the Oregon coast. We were left with 4 basic questions. Which species of pseudo are occurring off the coast? So if there's one, which one? And if there's several, which ones? Are different species occurring in different oceanographic conditions? So does the strength of the upwelling regime have an impact on which species occur or the proportions in which they occur? Then, regardless of species, is there seasonal or geographic variation in Pseudocalanus body size? And then if there is body size variation, is the species variation connected to the body size variation? So in order to answer these questions, I conducted a study that had 4 phases. In the first, I was identifying pseudocolonists in bulk plankton samples. In the second, I was measuring individual pseudocolonists. In the third, I was sequencing a gene from those individual pseudocolonists in order to identify what species they were. And in the final stage, I was analyzing both the genetic and body size data that I had collected for trends. So the samples I was working with were from 3 sites. I had a sample from La Push, Washington, and a sample from Trinidad, California to give some latitudinal diversity to my data. And then I had several samples from sites along the Newport line, and some of those sites I had samples from several months. So we had, all of these samples were from fall of 2018 through to early summer of this year. So we had seasonal resolution, north to south resolution, and inshore, offshore resolution. All of my samples were collected in vertical nets like the one pictured there. They're preserved in 95% ethanol, and I was identifying pseudocolonists under a dissection scope and just pulling them out with forceps. And that's what mixed copepods look like under the scope. So once I had pulled out my individual pseudocolonists, I would measure them, both their total length and also just the length of their prosome or body and their urosome or tail, because there was some thought that the ratio of those 2 measurements could be diagnostic as to species. In total, I measured 284 individual pseudocolonists for sequencing, which took a while. Once I finished measuring all of them, you can see they're all stored in tubes up there. I extracted DNA from them, amplified our target gene, which was CO1 or cytochrome C oxidase subunit 1, ran gels to make sure we had actually generated a product, and then took that amplified product, threw it in the sequencer, and received these raw sequences. Once I had both the body size data and the sequence data, I had 2 kind of parallel analytic workflows. For my sequences, I was loading the raw sequences into a program called Sequencer, where I aligned the sequences, trimmed them, and cleaned them, and then took those cleaned sequences and put them into a program called Mega, where I identified species. For the body sizes, I took my handwritten body size data sheets, typed them into Excel, and then did some statistics on them in R. So here is an image of this reference tree that I generated in Mega. I took reference sequences of CO1 for 4 species that we thought could be the pseudocolonist species we had off the coast, and built them into this reference tree. They were pseudocolonist minus, pseudocolonist pneumoni, minutus, and multoni. And then in order to figure out what species of pseudocolonists we had, I just took the clean sequences that I had generated and loaded them into the same tree. And the result was they were, without exception, pseudocolonist minus, which is kind of interesting because we had seasonal size variation and also some latitudinal variation, and that all of those organisms were the same species, which means that that body size was not a species difference signal that we were seeing. And just because we didn't see species variation, doesn't mean we didn't see any variation. There was body size variation on a couple of interesting axes. The first is the temporal. We were able to see a seasonal body size signal. This is just within my data. You can see in September, during the cold water high production regime, there are statistically significantly larger body sizes than there were in December, February, and April, which were during the warm water regime. And just, if anyone's not comfortable with these box plots, the dark line represents the median, the box represents the interquartiles, and the whiskers represent the extremes of the individuals measured in the sample. We also have access to a much larger data set because the Newport line has been sampled since 1996. There are pseudocolonists that have been measured since that year from samples taken every 2 weeks. So this is a data set collected largely by Dr. Bill Peterson, which has measurements for more than 16,000 individual pseudocolonists. And in this, you can see a really clear seasonal signal, which is pretty exciting. You can also see interannual variation that shows some larger than annual cycles. This is a chart of body size by year, and you can pick up some interesting oceanographic events, including the 1998 El Nino and a 2016 warming anomaly that was colloquially called the warm blob that occurred off the Pacific coast as well. We were also looking for spatial variation. We didn't see any statistically significant variation in body size with latitude, but we did see statistically significant variation in body size with distance offshore. So these are all samples taken within days of each other in September of last year along the Newport line from 525 and 65 miles offshore, respectively, and you can see they're all significantly different from one another, which is interesting. So our takeaways here are that the pseudocalanus found off of the Oregon coast in this last year were predominantly or exclusively pseudocalanus minus. We should take that with a grain of salt because, as we also saw, there's a pretty substantial signal of interannual variation, which means we likely haven't captured the full scope of variation that occurs. We also found that the pseudocalanus body size is varying through the year and also in between years, and we found that pseudocalanus body size varies with the distance offshore that samples are taken. So next, there's some next steps that we can take with this information. One thing I'd really be interested to do is further leverage the existing long-term data set that we have. Since we have this amazing time series that's at fairly high resolution going back to 1996, we could do a more quantitative analysis to look at the PBO or ENSO and see if we can pick up those signals and learn how they're impacting pseudocalanus body size. And we can also collect pseudocalanus from more sites, both so we can get better resolution on the question of how latitude affects body size, and also so we can begin to build a parallel genetic time series, which can help us to understand species variation along with body size variation. I have a few people who I really need to thank. The most important are my mentors, Kim Jacobson, Dave Jacobson, and Michael Banks, who took amazing care of me this summer and taught me a ton. I also have to thank a bunch of other people who provided me with data, helped me with bioinformatics, helped me generate figures or gave me figures, and helped me just talk through problems I was having with the project. I'd like to give a special shout out to Mark DeSimone, who's presenting on this very stage later today, so keep an eye out for him. Here are some sources, and now I'll be happy to take your questions, and if we can't get to any, email me.
Was the zooplankton community made up entirely of pseudocalanus, or were there other species as well?
So there are other species in the high production. So in the high production regime, there's relatively few copepods that occur, and they occur in great abundance. The most common ones are pseudocalanus, Calanus marshalli, and Carchalongeramus. In the warmer water regime, you have much more copepod diversity and much lower abundance of pseudocalanus. And other non-copepod zooplankton also occur.
What was the gene that was sequenced, or what is it coded for?
The gene that was sequenced is cytochrome C oxidase subunit 1. It's a mitochondrial gene that is used in a lot of different barcoding studies because it has great coverage across species and also varies a lot between species. It is involved in energy metabolism. So pseudocalanus are super small. They're between 1 and 2 millimeters in total length. Ours were preserved in ethanol, so I had them in ethanol, crushed them with a pipette tip, and then did a fairly standard tissue extraction with sequential chloroformances and that kind of thing. I think it's part of that is because upwelling is way stronger closer to shore. As you can kind of see in this first figure, just a sea surface temperature. So you have like a much stronger gradient with distance moving offshore than you do moving down the coast. But I do think it's an interesting question and worth further study. Thank you.